Okay, first, thank you for the invitation. Thank for everyone for connecting. Today, we'll talk about the Notch3 positive regulatory T cell that drive autoimmune neuroinflammation in multiple sclerosis. As a bit of an introduction, natural Treg is developed in the thymus during the T cell development, where immature thymocytes go from positive selection to negative selection, where the cell with high affinity for the TCR are deleted by apoptosis, and the cell with an intermediate affinity become natural Treg progenitor. The cell with a low affinity become mature naive T cells. After those selection, these both cell type migrate to the periphery, where Tregs will control and induce immune tolerance by controlling cells that escape negative selection and become autoreactive in periphery, but also all type of inflammatory process. In parallel, naive T cells can also become Tregs in periphery by acquiring the expression of FOXP3 that are the master regulator of the Treg lineage, and it's what we call the peripheral Treg. And those Treg will also help to control this inflammatory process or autoimmune inflammation. Based on all that and this central role of regulatory T cell in immune tolerance, several labs and companies try to use Tregs as a therapeutic approach by first using a genomic engineering strategy, and to do a long story short, by trying to express FOXP3 in naive T cell or memory T cells to let them acquire immune suppressive mechanisms. However, this approach is not the best one because FOXP3 alone is not enough to become regulatory T cells. Another strategy was to expand in vivo regulatory T cell by targeting specifically CD25 that are the high affinity receptor of IL-2. And the idea was to use low dose IL-2, like IL-2 mutein or IL-2 anti-IL-2 complex, to expand specifically regulatory T cell in vivo. And the last approach is adoptive cell therapy with generating CAR Tregs. However, one of the major limitations of all this approach is a lack of consideration of the tissue Treg heterogeneity. Indeed, since a couple of years, it was shown that tissue Treg are really specific transcriptionically speaking. As you can see in this PC analysis, they are all different one to each other, and they are also different from circulating Tregs or Treg present in the lymphoid tissue. To go more in detail, it was shown that tissue Treg express a specific pan-tissue Treg transcriptome, as you can see here. They all have the same. However, each tissue Treg from each tissue present a specific transcriptome. In another word, Treg from the skin will not express the same transcriptomes as Treg from the meninges or the adipose tissue. Based on that, and in line with this idea, in the lab since a couple of years, we work on the Notch signaling, and we try to understand the role of Notch signaling in Treg cell-mediated inflammation. The Notch signaling is composed of 4 receptors and 5 ligands, and in the lab, we focus on this signaling in Tregs. First, a couple of years ago, we show in a lung inflammation, first in an allergic airway inflammation, we show that Tregs will upregulate Notch4, and this upregulation will lead to a destabilization of Treg and leading to an exacerbation of lung inflammation. To go further, we confirm that in another lung inflammation model, a lung viral infection, where also in this model, Notch4 is upregulated in Treg, and this will lead to an exacerbation of lung inflammation. However, the mechanisms that induce this exacerbation in viral infection is different than the mechanisms that we can find in allergic airway inflammation. More recently, we have shown that another Notch receptor, Notch1, can be also upregulated in Tregs during another type of disease, and this time was in a multi-inflammatory syndrome. And what we've shown is that this upregulation will lead to a loss of FOXP3 expression and an exacerbation of the systemic inflammation. Based on all these results, we came with an hypothesis that each Notch receptor will control or participate to a tissue-specific inflammation. And based on that, we wanted to know if the Notch signaling drives Treg dysfunction in CNS inflammation. We decided to focus on multiple sclerosis because it was shown that in MS patients and in a mouse model of MS, the EAE model, Treg are defective and pathogenic. However, the mechanism that drives this dysfunction and this pathogenicity in MS and EAE was unknown. And we came with the hypothesis that maybe the Notch signaling play a role in this dysfunction. Thanks to a collaboration with Violeta Jujevic in Paris and Raffaele De Palma in Italy, we had access to MS patient, and we analyzed first like the expression of the different Notch receptor in the circulation. What we find, and as you can see here in this FACS plot and this quantification, is that the only Notch receptors that are upregulated in MS patient are Notch3, sorry. And specifically in Treg, because the T cells that don't express FOXP3 are not expressing like any Notch. To go further, we had access to cerebrospinal fluid of this patient, and we also analyzed the expression of Notch3 in Treg on those cerebrospinal fluid samples. What we find is that in the cerebrospinal fluid, Notch3's Tregs are like Notch3 is expressed highly in this compared to the peripheral blood of the same patient. Finally, as I think you know, like MS, you have different type of MS, and the more prevalent MS is a relapse-remitting disease, where patients go from an active phase to an inactive phase of the disease. And we analyze the expression of Notch3 during this both phase of the disease. What we find is that Notch3 is found in the peripheral blood of patients only during the active phase of the disease. This result pushes us to think about the role and an active role of Notch3 in this disease, that Notch3 participate or induce the disease in MS. To analyze this, we decide to switch to a mouse model, and we decide to use the EAE mouse model that consists of immunized mice with CFA and the mock peptide 3555. And at D0 and D2, we inject with pertussis toxin. After that, the mice will develop a progressive paralysis going from after 8 to 10 days, from a score going from 0 to 4. First, we try to confirm that we find the same upregulation of Notch3 in this disease. We analyze at the peak of the disease the expression of the different Notch receptor in the infiltrating CD4 cell in the CNS. What we find is, as in human, Notch3 is the only Notch receptors that are induced during EAE in the CNS. To really analyze the role of Notch3 in this disease, we decide to deplete Notch3 specifically in Treg by using the FOXP3 or FP3 mice. And as you can see, the deletion of Notch3 prevents decrease the EAE severity. And this is specific of Notch3 because the deletion of the other Notch receptor are not inducing the same protection. To go further, we analyze the production of pro-inflammatory cytokine by infiltrating CD4 cells. And what we find is that the protection induced by Notch3 deletion in Treg is associated with a decrease of pro-inflammatory cytokine production by infiltrating CD4 cells, and mainly the cells that are producing IL-17 and GM-CSF. To go further, we decide to characterize this population, like the Notch3 positive Treg population in the CNS at the peak of the disease. And we did an immune phenotyping analysis. First, what we find is that the Notch3 positive Treg express a lower level of FOXP3 in the CNS. And this is associated with an activated phenotype, as you can see with this upregulation of CD69 and CD44. To go further, we decide to analyze the antigen specificity of these cells, and we analyze with a MOX staining, tetramer staining, the MOX specificity. What we find is that the Notch3 positive Treg and FOXP3 negative Notch3 positive cells express a recognized MOG peptide compared to the Notch3 negative cells. To go further and to go fast on the long story, we analyze several markers, and what we find is that the Notch3 positive Tregs express alpha-4 beta-7, that is a gut-associated marker and a gut homing marker. Several studies have shown that the gut microbiota and the gut-brain axis is really involved in MS and EAE. And based on that, we wanted to know that if the gut microbiota is involved in the induction of Notch3 expression. For that, we first analyze at steady state the expression of Notch3 in the lamina propria, the gut lamina propria of the mice. And we find that at steady states, Tregs express Notch3 in the gut. To go further, we decide to perform the same analysis in the germ-free mice that have no macrobiota. And we show that by removing the macrobiota, Notch3 is not expressed in Tregs. To really confirm that the macrobiota is involved in this induction, we decide to do a fecal matter transplant to transfer macrobiota from the SPS mice to the germ-free. What we realize is after the transfer of macrobiota in a germ-free mice, Notch3 expression is restored in the gut, but also in the CNS, really showing that the gut microbiota is involved in Notch3 expression in the gut. To go further and to really understand if the Notch3 positive Tregs are the gut Notch3 positive Treg are migrating to the CNS, we decide to block the gut homing or the CNS homing by using a specific antibody blocking experiment. We induce an EAE in a wild-type mice, and we treat the mice with an anti-alpha 4 beta 7 to block the gut homing or an anti-VLF4 to block the CNS homing every 48 hours. What we find at the peak of the disease at day 15 is that the Notch3 expression is normal as what we found before in the isotype control. However, if we block the gut homing or the CNS homing, we lost the expression of Notch3 in the CNS, really confirming that the gut Tregs or the migration to the gut and the acquisition of Notch3 in the gut is necessary to find the Notch3 Treg in the CNS. To conclude on this part, what we have shown is that the microbiota, through a TLR-dependent mechanisms, will induce Notch3 in the CNS, in the gut. This Notch3 positive Treg will migrate to the CNS through an integrin-dependent mechanisms. The next part of this work was to understand what those cells will do in the CNS during EA. To this end, we decided to do a single-cell RNA-seq in the wild-type or the Notch3 specific, the Treg-specific Notch3 deletion in Tregs. And we find that 12 clusters, as you can see here, and what we find when we did our analysis, cell proportion analysis, only 2 clusters was proportionally different. The cluster 11 that represent a naive CD4 T cells cluster, and the cluster 7 that present a mixed Treg Th17 phenotype. We decided to focus on this cluster because it was shown that during inflammatory process, Treg can be destabilized and acquire the capacity to produce pro-inflammatory cytokine. So what we call Treg destabilization and also X-Treg generation. We decided to test the X-Treg generation in our model. For that, we generate a mice to target and to track the X-Treg in vivo. We cross the FOXP3 YFP3 mice with a ROSA26 lock-stop-lox RFP. In this mice, your Treg will express YFP RFP, and when they will lose FOXP3 expression, they will be only RFP positive. We cross these mice with a Notch3 flux, and we induce EA. What we find at the peak of the disease is in a wild-type mice, we have an increase of X-Treg generation, as you can see here in black. However, if you delete Notch3, you prevent or decrease this generation of X-Treg, already showing that Notch3 expression in Treg lead to X-Treg generation and Treg destabilization in the CNS. To go further and to really characterize this population of X-Treg, we decide to analyze if this X-Treg produce pro-inflammatory cytokine. What we did, we check and analyze in the cells that are producing, all CD4 cells that are producing interferon gamma, or interferon gamma IL-17, IL-17, or GM-CSF, the contribution of T conventional, Treg, and X-Treg. For example, for the interferon gamma here, 82% of the interferon gamma that are produced in the CNS during EAE come from T conventional. If you check the Treg is around 9%, and the X-Treg about around 8%. Here is the quantification of all our mice. We did the same with the interferon gamma IL-17 double positive, and here we see that 30% of this population, of this both cytokine or cells that are producing both cytokine are X-Tregs. And we did the same with IL-17 alone, and is around 15% of the IL-17 produced in the CNS during EAE are produced by X-Treg. We quantify all that, and what we find is that the X-Treg produce around 15 to 20% of interferon gamma during EAE. This was already previously published by the lab of Bluestone a couple of years ago. But what we find in our study is that around 30 to 40% of the cells of the X-Treg, 30 or 40% of the L17 is produced by X-Treg, sorry. And we did the same for GM-CSF, and around 5% of the GM-CSF are produced by X-Treg. To go further and to really characterize this destabilization and to understand how this process happened, we wanted to know first where this happened. It is in the gut or in the CNS. What we did at the peak of the EA disease, we analyzed the X-Treg generation in different tissue, brain, spinal cord, meninges, gut lamina propria, and spleen. And as you can see here, the Treg destabilization happened mainly and really only in the CNS, because in the gut and spleen, you have the same level of what you can find in a PBS mice. To go further, we wanted to understand what are the ligands and which cells express the Notch ligand to induce the signal in Tregs. We did a lot of analysis, but at the end, we focused on the macroglia, and we find that in the macroglia during EAE, you have an upregulation of DLL1 specifically, both in a wild-type background or in the FOXP2-FP3 Notch3 flox mice. To really analyze the impact of the role of DLL1 in this destabilization, we first did it in vivo by using an antibody against DLL1. And we find that blocking DLL1 during EAE induces the same protection that deleting Notch3 in Tregs. And this is also associated with a decrease of Treg generation. To really confirm that it's a really active interaction between DLL1 and Notch3, we went to an in vitro model where at the peak of the disease, we isolate Tregs from wild-type background or Notch3 deficient background, and we co-cultive them with macroglia pulse with mock peptide. First, what we find is if you delete Notch3 in this culture after 3 days, you have less destabilization of the Treg, as you can see here with the FOXP3 expression. So we have normally here FOXP3. What we did, we use the same antibody, the blocking antibody against DLL1, and we show in a wild-type background, we prevent this destabilization. And as a control, we use an anti-Jagged2, and these anti-Jagged2 are not inducing the same protection. Here is a quantification of really showing that you prevent the destabilization with anti-DLL1, but not with anti-Jagged2. And in a Notch3 background, blocking anti-DLL1 are not changing, really showing that after the migration to the CNS, the Notch3 positive Treg will interact with macroglia that express DLL1 to induce this destabilization. The last part of this destabilization, we wanted to understand which signaling inside the Treg are necessary or inducing this destabilization. To do a long story short, in our single-cell RNA-seq analysis, we did a pseudo-bulking of our cluster 7, and we find one of the top pathways that are differentially expressed between wild-type and Notch3 deficient Treg are the Hippo pathway. The Hippo pathway is induced by two specific molecules, YAP1 and TAZ, and we decide to analyze the expression of YAP1 at day 15 after EA induction. What we find is that deleting Notch3 in Treg prevents the upregulation of YAP1. To go further, we decide to delete the Hippo pathway completely in Treg by deleting YAP and TAZ in Treg. And as you can see here in this analysis, this induces the same protection of the Notch3 the deficiency in Treg, and this also prevents the generation of Tregs. To really confirm that, we also use the same approach, in vitro approach that we showed before, and we really show that deleting Notch3 or YAP-TAZ in Treg prevents the destabilization in vitro after a co-culture with wild-type macroglia pulse with mock peptide. To conclude on this both part, what we have shown in this study is that macrobiota induce Notch3 expression in Treg in the gut. This Notch3 positive Treg migrate to the CNS through integrin-dependent mechanisms. When they arrive in the CNS, they will interact with macroglia expressing DLL1. This will lead to an activation of YAP-TAZ in Treg, leading to their destabilization and the generation of Tregs producing IL-17. And this population represents 30 to 40% of the cells that produce IL-17 during EA. What was interesting in this study is, like in our transcriptomic analysis, we find that deleting specifically Notch3 in Treg induce the upregulation of a neuropeptide receptor, the neuropeptide Y1 receptor. First, we confirm this upregulation and the expression of this marker in Treg in the CNS by flow cytometry. And as you can see, at steady state, neuropeptide Y1 receptor is expressed specifically in the CNS. You don't find it in the spleen or in the gut. And it's really expressed in the brain, spinal cord, and meninges. We did several analyses, and we realized that during EAE, this population decreased. As you can see, if you compare the light blue to like the dark blue, and in contrast, this population decreased, but the Notch3 positive population increased in all parts of the CNS. Based on this change on this both population, we decide to characterize and to compare both population by flow cytometry. And what we find is that the neuropeptide Y1 receptor express a higher level of FOXP3 helios, a lower level of gut homing marker, alpha-4 beta-7. And to do a long story short, we analyze several markers in our bulk RNA-seq, single cell, and so on. And we confirm that these cells express specific CNS resident or tissue resident marker. As you can see with this upregulation of CD103, like the neuropeptide Y1 receptor are mainly double positive, also the expression of higher expression of CD69, and they express a lower level of CD41D, 49D that is involved in the migration to the CNS. To finish this study, we really wanted to know that if this population play a role in EAE, and we decided to delete it in Tregs by using the FOXP3 mice. What we find is that the deletion of this gene in Treg induces a rapid, severe lethal EAE, as you can see here in pink, and also in the survival curve. We also analyze the impact of this population on the cytokine production by T conventional or CD4 cell infiltrating the CNS. And what we find is that like the specific deletion of neuropeptide 1 receptor in Treg drive interferon gamma and the MCSF producing T cells, as you can see here in blue. To conclude on all this study, what we have shown that is gut microbiota and like will induce Notch3 positive, Notch3 expression in Treg in the gut through TLR-dependent mechanisms. Those cells will migrate to the CNS thanks to an integrin-dependent mechanisms. When they will arrive in the CNS, they will interact with microglia that express DLL1. This will lead to an activation of the EPO pathway, leading to the Treg destabilization and the acquisition of a next Treg Th17 phenotype that will contribute to around 30 to 40% of the whole IL-17 producing in the CNS during EA. In parallel, this destabilization will outcompete a Treg CNS resident population characterized by neuropeptide 1 receptor that normally control the interferon gamma and the GM-CSF. With all that, I would like to thank